Tēnā koutou katoa. Welcome to the third day of the Australasian Epidemiology Association annual scientific meeting. And we're really delighted to have this morning's session having a strong emphasis on the links between human and animal disease. And as many of you are aware, the One Health approach has a huge amount to offer both human and veterinary epidemiology. And we're really delighted to have Professor Nigel French as the opening speaker this morning, and he's going to speak on controlling zoonoses, the increasing role played by molecular and genomic epidemiology. And I've had a lot of contact and collaboration with Nigel for a number of years, and each time I'm absolutely staggered by the quality of veterinary epidemiology at Massey, and I think they're absolutely a world centre in this area. And I think that in the human epidemiology area, I think we can learn a huge amount from the approaches they take. And I think Nigel's also remarkable in the breadth of his use of different methods from epidemiological, spatial, and molecular epidemiology, and just synthesizing all of that. And I think it really shows some of the best of a multidisciplinary approach to epidemiology. And as we'll hear in the second speaker in this session, I also think the global approach and the social responsibility that's built into these approaches. You've probably seen Nigel's extensive bio sketch in the program, but he's Professor of Food Safety and Veterinary Public Health at Massey University, Director of the Infectious Disease Research Centre, Head of Molecular Epidemiology in the Public Health Laboratory at the Hopkirk Centre. And he's really done, I think, extremely important work in New Zealand that's really changed the face of zoonotic disease in New Zealand. And he has also answered the so what aspect by linking his work to actually making a difference to disease burden. And so I think we're in for a fantastic session. So I'll hand over to Nigel at this point.
Well, thank you very much, Michael, and good morning, everyone. And thanks very much for giving me the opportunity to talk at this conference. It's the first time I've been to a meeting of this kind, and it's a real privilege to be here. So the title of my talk, I'm going to be talking about a little bit of an introduction around zoonotic disease generally and some of the big issues. There's obviously some major issues ongoing at the moment in the world related to zoonotic disease. Then I'm going to focus a bit on how we've applied molecular epidemiological tools to tackle foodborne disease in New Zealand, and giving the case study of Campylobacter infection. And then lastly, the last third of the talk will be around the use of new next-generation sequencing tools and the sequencing of whole genomes of microorganisms to inform epidemiology and the control of infectious disease. And that focus at the end will be on Salmonella in New Zealand. So my laboratory is based in the Hopkirk Institute, which if you've ever been there to Massey, Palmerston North, it's actually linked to the veterinary school. One of the 6 laboratories in the building is the MEPI lab. And we have a One Health focus in there. We focus on food safety, water quality. We develop tools for surveillance, space-time tools. We're actually involved in vaccination programs for measles at the moment with the Ministry of Health. We have one of the world's experts on Ebola in the team too, which is very topical. And we also, molecular epidemiology of zoonoses, and those are a list of the kinds of bugs that we're working on at the moment. And it's one of the 6 groups that form the Infectious Disease Research Centre. And if you're interested in the kind of work that the centre does, there's a website there that you can have a look at, which will give you an update of the kind of research programs, the publications that are coming out of that particular group. And that really is One Health in its broadest context, because it also includes plant infectious disease. So zoonotic disease, or zoonoses, so these are diseases that are transmitted between animals and man, and the between is an important word there because you can have reverse zoonoses, so you can have reverse infections from humans back into animals, and probably what happens in major epidemics of zoonotic disease is you actually get transmission occurring both ways. It's just that some are more prevalent than others, so usually you get more animal to human, but you can certainly get it going in the other way. And it's characterized usually by animal reservoirs, so these could be both livestock and wildlife, very often wildlife reservoirs for maintaining the infections. Then you can have what are called bridging hosts, which can't maintain the infection themselves, but can be very important for transmitting between the reservoir or maintenance hosts and people. And it's pretty well known now that most human infectious diseases are zoonotic diseases. And it's not just the mild and insignificant ones. Obviously, there's some major ones as well. And probably more importantly, we see regularly new infectious diseases that emerge, and we see the resurgence of existing infectious diseases. And most of those diseases, 3/4 of those, are zoonoses. And this is obviously one good example, the Ebola outbreak that's going on in West Africa at the moment. A couple of other features is that about a third of these zoonoses don't just rely on the animal host, they then become transmissible between people, and I'll expand on that in a second. And another important feature is many, probably most, zoonotic diseases have minimal effect on their animal host, and that becomes very important when you're trying to control the reservoir infection. Often there's very little demand, particularly if it's a livestock-associated infection, to vaccinate because it simply doesn't cause production effects in the animals. So zoonoses can be epidemics, they can be pandemics, so obviously they can be very important from a global perspective. SARS, Ebola, bird flu, swine flu, Nipah virus, major burdens to the health services and economy of many countries around the world. These outbreaks are very important, but it's also worth bearing in mind that still around about 50,000 people a year die from highly preventable diseases such as rabies. I've just given you a few examples at the bottom of zoonoses just associated with cows and sheep. So we have the sugar toxigenic E. coli 0157, we have salmonella strains, obviously CJD, leptospirosis, which will be talked about later, and a number of other diseases as well. And we should be reminded that the three major pandemics that caused such a dent on the human population throughout history, bubonic plague, the Spanish flu, and HIV, are all originated as zoonotic diseases. And Spanish flu certainly had a major impact on New Zealand. This is a picture here of private cars being used as ambulances during the epidemic in 1918. And this was a headline in the newspaper this morning, so highly topical, just across the ditch. I don't think it is, but no doubt we'll find out soon. So really what we're talking about are multi-host pathogens, and there are a huge number of multi-host pathogens, of which zoonoses, the ones that go between humans and animals, are a subset of those. So single-host pathogens are relatively rare, and of those, the ones that transmitted between people alone are called the anthrapanoses, and it's believed that those only probably really evolved and started circulating in human populations once human populations began to reach certain sizes and began to coalesce in areas such that the fuel was enough to maintain infection. There are various different stages that emerging zoonotic diseases go through, and some of them seem to be stuck, like rabies here, where your primary infection you need the animal reservoir, and humans only get infected from animals. At the other end of the extreme, even though it's relatively recently emerged, the two HIV strains are primarily now spread between people. So they've moved into a stage, what's called a stage 5 zoonotic pathogen. I think Ebola has probably moved from being a stage 3 pathogen with few and limited cycles of the current epidemic into a stage 4 pathogen. So why are we seeing this increase? There's a whole number of features that are contributing to this. Many of them are well known. What we've got here, you've probably seen this picture many times, but it's the flight paths of aeroplanes going across from North America back to Europe, and then over a 24-hour period you see them streaming back over the other side. So the increased movement of people, of animals, of food around the world is almost certainly contributing to the dissemination of emerged pathogens. And other factors such as the encroachment and urbanization, encroachment of agriculture into wildlife areas is almost certainly contributing to the emergence of new pathogens as well. So there are many factors, including climate change, and habitat destruction, that are contributing to this emergence and resurgence of new pathogens. So what about New Zealand? We do pretty well in terms of zoonotic pathogens. I'm talking about well in terms of if you're researching zoonotic pathogens, New Zealand's a fantastic place to come, because we actually have some of the highest rates of many of the zoonotic pathogens, if not the highest. And they have a whole range of different pathways by which we can contract them. So we take Campylobacter on the right, which I'm going to talk about a little bit more about. We have about 8,000 cases a year, probably mostly still food spread. We have Cryptosporidium and Giardia, around about 1,000 cases per year. Those are mainly waterborne, recreation and swimming, and drinking water infections. We're amongst the highest rates of those in the world, probably second highest in the world for this. Salmonella, I'm going to talk a little bit more about this infection, which almost certainly came from wild birds originally, caused a massive epidemic in New Zealand. And then we have the sugar toxigenic E. coli. We're around about 250 cases a year now, and I think we're second in the world behind Argentina. So a great place to work on zoonotic disease. And leptospirosis is occupationally acquired, and Cord Hoyer will be talking more about that later. So a little bit of a focus on foodborne zoonoses in New Zealand. So this is a story about Campylobacter. Campylobacter is still top of the list, measured in order of magnitude higher than many of the other notifiable diseases in New Zealand, certainly highest of the enteric notifiable diseases. You can see the other ones starred here are the zoonoses, so it really highlights how important zoonoses are in terms of New Zealand and notifiable diseases. For those of you, you're probably all aware of this, but it's mainly two different species of Campylobacter. Campylobacter jejuni is the predominant one, causes about 90% of cases. Those are the clinical signs that you see associated with infection. It's estimated around about 1 in 15 are hospitalized, and you can get these really serious complications, the progressive neuropathies, such as Guillain-Barré syndrome. But mainly it's the bottom dropped out of your world and the world dropped out of your bottom feeling, which you get, and it's pretty serious over a period of a year, over a week or so. Sorry, not a year. And really what we were faced with was a major and growing epidemic of Campylobacter right up to the year 2006. And this is, it really illustrates how New Zealand fared compared to the rest of the world. And before I moved to New Zealand in 2004, and New Zealand was known as the Campylobacter capital of the world. And if you ever wanted to work on Campylobacter, this was the right place to come. And for obvious reasons. And if you actually look at a map, so this is a map of over 100,000 cases of Campylobacter over the period of 10 years between 2000 to 2010. This is mapped to the mesh block level using a little bit of software called Cases to KML, which you can automatically download onto Google Earth Maps. The right side is obviously Wellington, the left is the rest of New Zealand. And this is really a map of misery, indicating that Campylobacter was visible from space. And this was a very important paper that came out in 2006. You can see Michael Baker was the first author on this paper. And this was really a major attempt to try and get something done. There was a lot of evidence, growing evidence from previous studies, that poultry were primarily responsible for human infection. But nevertheless, despite all the efforts and all the previous work that had been going on, there was still a lot of uncertainty in certain quarters, particularly, I have to say, in the poultry industry. And what was contributing to the uncertainty, or what was being used by industry at the time, was the fact that some of the previous studies appeared to have contradicting findings, such as if you ate poultry at a friend's house, this is from a case control study, you were 3 times more likely to get Campylobacter, whereas if you ate it at home, you were 3 times less likely. And it was that kind of confusion, coupled with other things that were appearing around about the same time, which were suggesting that maybe it was cows and sheep and contaminated drinking water supplies that were primarily responsible for the big number of cases of Campylobacter. So it took a concerted effort building on those previous approaches and adding a new method, I think, to try and convince the poultry industry that they really needed to comply with the risk management programs which were being put in place by the Food Safety Authority at the time. So the approach that was taken then was what's called source attribution, or reservoir attribution as it's also called. And that's really fundamentally trying to identify what are the primary cause sources of human infection. So where are people getting infected from? So it's a surveillance activity. You can use it as a monitoring activity post-intervention, and those are the fundamental questions that we're asking. And this is from a paper in Clinical Infectious Diseases, but also was part of a framework put together for the World Health Organization around this whole area of source attribution and how you combine some of the modeling work of reservoir attribution, trying to identify what the primary reservoirs for infection, through to understanding what then feeds into different pathways, such as food chain, direct contact, drinking and recreational swimming, how you relate that to individual case control studies, for example, where you're identifying very proximal risk factors within that framework to understand the epidemiology of these infections to inform policy for control. Now, we took an approach, a molecular epidemiological approach, and the approach that we took really captured some of the techniques that were just emerging at the time, around about 2000, the mid-2000s. And one of the first things we did was set up the Manawatu region as a sentinel surveillance site. So the Manawatu, for those of you who don't know, is just located here. It's right next to the university where I work, so my laboratory is right in the middle of that region. And what we did was all the human clinical cases that were confirmed by ELISA came through to our laboratory. We tested those, and we also sampled food from a whole range of different retail outlets in town and in other areas as well. We sampled high-use recreational swimming spots, and we sampled farms from the catchment areas of those swimming spots, all in the same space, all in the same time, using exactly the same culture techniques. We then used a technique called multilocus sequence typing, which had recently emerged as a very useful typing technique for Campylobacter, and it was useful because it really helped to identify source. So a very strong association between sequence type and source for many of the different types that we had. I won't go into the details of this, but just to say it was a very useful technique, and there was an international database which was set up, which was great for comparing isolates that we had from New Zealand with those for the rest of the world. Now this slide, I call it the beach ball slide because it can be a bit tricky, but basically each of those circles is a sequence type or a genotype of Campylobacter. The size of the circle represents how many isolates we've had of that particular type, and the pie chart within that circle represents where we isolated that particular type from. And how they're arranged on the screen here, if there's a strong black line, it suggests they're very closely related to each other. The further they are away from each other, the more distantly related those Campylobacter isolates are. And one of the things that emerged very early on was that this particular sequence type, 474, very rare anywhere else in the world, was the most dominant sequence type in New Zealand. And more importantly, it came from one particular poultry supplier, particularly in the early years of this infection, this study. We can also see there are ruminant-associated genotypes here, here, and here, and some that are just found in recreational swimming water as well. So what we needed to do was to take that information and then use it in an epidemiological setting. And one of the first things we could do was now to look at maps of Campylobacter, but by sequence type or by host-associated sequence type. And one of the things that emerged fairly early on using these techniques of spatial modeling was that the sequence type 474, the one associated with the poultry supply, was predominantly an urban-associated infection, predominantly found high risk in Palmerston North, in the centre of Palmerston North, and relatively low risk in the rural areas surrounding. Whereas if we took the ruminant-associated sequence types, we found the opposite picture. Green is low relative risk, red is high relative risk, that was predominantly found in those rural areas. So that was telling us something about the likely types of exposure that we were seeing. Now, around about the same time as well, a paper was published by Danny Wilson, which described a new technique for combining multilocus sequence type information with evolutionary modeling to infer where humans were getting infected from. It's a population genetics approach. It basically reconstructs the genealogical history going back through time of the Campylobacter isolates, estimates mutation, recombination rates, but most importantly, from a public health perspective, the relative migration from what we'll call islands. It's called the asymmetric island model. Each island is a host reservoir into the human population. It was these relative rates of migration that gave us the estimates of where humans were getting infected from. Now, the answer that came fairly strongly from this study, and we used two other models as well, which were both published in a comparative way, was that poultry indeed were the predominant source. Actually, one particular poultry supplier appeared to be counting for most of the cases in the Manawatu region. There was a contribution from cows and sheep, but really it was poultry. So that was then useful for informing the Food Safety Authority. They were able to implement their Campylobacter risk management strategy. The poultry industry complied, opened their doors to each other, and really worked hard to try and reduce the level of contamination in their supply. There was no single intervention. A whole range of different things were done, mainly around this thing here, which is a spin chiller. If you've ever been in a poultry slaughter plant, the spin chiller, basically the birds are killed, eviscerated, plucked, and then they're at body temperature. They have to try and get them down to cooling temperature before they put in the bag and sent to the supermarket. And really what these spin chillers were doing was creating a kind of fecal soup, and the chickens are rotated in that fecal soup and then bagged up and sent through to the supermarket. Huge improvements in that area around pH control, filtration, really helped to reduce the contamination of those chickens. But the first information that came through around the levels of contamination in chickens was through the National Microbiological Database, which suggested initially a relatively modest reduction in the level of hazard in chicken carcasses going through into the supermarket. But a 0.25 reduction in here actually corresponds to a 50% reduction in real terms because it's on a log scale. But the real interesting thing was what happened to public health. And I think it really took us by surprise, the impact on public health from those interventions, or what coincided with that was a massive drop in clinical cases or notified cases of around about 50%, and that also corresponded with a 50% reduction in hospitalization cases. This is from a paper by Anne Sears in Emerging Infectious Disease, and Michael and I are both co-authors and were involved in the supervision of Anne in this work. And it's estimated that this reduction alone accounts for around about a $50 to $70 million per annum saving to the New Zealand economy in recent economic estimates that have been done. And it has persisted through to the present day, so even now we're around about 8,000 cases. Shouldn't be complacent, obviously. And this departure occurred around about 2007. You still see that seasonality that's occurring there. We've got dynamic reservoir attribution models based on the coalescent model, which I described earlier, which we use for monitoring purposes for the food safety people in the Ministry of Primary Industries, and which really show how the bulk of the reduction occurred as a result of reduction in poultry contamination. And this big reduction had a dramatic effect. It changed the epidemiology of Campylobacter in New Zealand completely. You can see prior to the intervention in the poultry industry, most cases were in urban areas, or the highest rates and risks were in urban areas. But after the intervention, it swapped over, and now the highest rates are actually in preschool children in rural areas of New Zealand, and this is right the way across the whole of New Zealand. So, what's happening in those rural areas? This is just from the 2013. We actually attribution estimates for the Sentinel site, and if you break it down into rural and urban cases, you can see that in urban areas, the predominant source still seems to be poultry, but certainly in those rural areas, we're seeing ruminants as being the most important source of infection. Why are the different epidemiology? And this is where spatial mapping comes in, just taking the cases, all the notifiable cases, and comparing them with a map of livestock densities. And what we find when we do the modeling is that there's a very, very strong association with dairy farming density, not sheep farming or beef farming, but very strong association with dairy farming. The main dairy farming areas around the Waikato, Taranaki and South Canterbury appear to be areas where the highest incidence is occurring. Now we know, so where is it coming from? We know raw milk is a source of Campylobacter infections. More and more people drinking raw milk in New Zealand. More and more farmers selling raw milk, and more people getting sick, but it doesn't seem to be stopping people from drinking raw milk. But that didn't account for the increase in rural areas. These are mainly urban people that seem to be going and buying raw milk and consuming it and getting sick. So really what we get is a clue from looking at the time series of infection. In urban areas after the poultry intervention, we see a big decline in those urban areas as expected. In rural areas where there are no cattle, we see a similar pattern, although slightly lower in those early years. But when you look at the high-density dairy farming areas and look at the incidence over time, what you see is that after the poultry intervention, the infection rates remain high, but you may also notice that the peak in infections occur a few months before, in almost every year, before the peak in the urban areas and the areas with cattle. And just to summarize that figure in a box plot, what you see is this peak in the high-density dairy farming areas around about July, August, September time, which corresponds with the calving period. And it's probably infections of young children feeding calves, contamination. There's this strong lamb and calf days they have in rural areas in schools as well. So it's probably associated with those kinds of exposures. Okay, so move on to the last part of the talk. So that's I've talked about molecular epidemiological tools, and you'll notice that the tools I was using there, in combination with things like spatial mapping and modeling and all those sorts of things, really help to inform the Campylobacter control strategy. But really what's happened recently is a revolution in the way we do molecular epidemiology. with the advent of next-generation sequencing. We now have, instead of relatively low-resolution typing of our organisms, we have the ultimately high-resolution typing. We have full genome sequences. And so this new term has emerged, genomic epidemiology, increasingly being used around the world for infectious disease. It's also used for non-infectious disease as well. And it really is the application of genome sequencing to inform epidemiology in its simplest terms. You'll be familiar with genome-wide association studies. They're now being used for both communicable and non-communicable diseases. And the number of studies using these techniques has increased. But there's still a lot of methodological work that needs to be done to improve the way we apply these techniques. And recent papers and publications have come out on a whole range of different diseases, a whole range of applications, understanding population structure, transmission mechanisms, also the impact of interventions such as vaccination strategies, and a lot of publications in very high-profile journals such as Nature Science and PNAS appearing. There are a lot of review articles, if you're interested in as well, which are make very good reading. I recommend those two particular ones there by Pybus and Croucher if you're just interested in getting an overview of how these techniques are being applied. And in terms of how they're being applied, I'll come back to this slide right at the end just to summarize, but in essence, they're being applied to infer transmission rates, estimating the basic reproduction number, which can be used to inform vaccination strategies, how transmission's occurring amongst social networks of people and animals, inferring zoonotic transmission, host switching events, the relationship between pathogen genotype and phenotype, which is very useful for inferring the source of infection if you can get host-associated genotypes, inferring long-scale geographical movements of pathogens around the world, and the origin and introduction, very useful for biosecurity and the incursion control, and also for understanding the evolution of pathogens and the impact of interventions such as vaccination and antimicrobial resistance use on the emergence of resistant strains. And Debbie Williamson from ESR is doing some nice work on using genome sequencing tools to inform, from Otago and ESR, to inform control of antimicrobial resistance. A nice example, actually not a zoonotic example, was the New England Journal of Medicine one. It's a relatively old one, because it's 2011. A 3-year outbreak of TB, traditional contact tracing combined with the low-resolution genotyping that was around at the time, a technique called VNTR, really wasn't very helpful because it just identified a single clone. There was no discrimination. Whole genome sequencing, in contrast, identified that not was there one outbreak, it was actually 2 concomitant outbreaks occurring at the same time. When they combined that with the social network analysis, they identified that actually one of those 2 outbreaks that was happening concomitantly was caused by one particular superspreader individual. And that helped to inform the much better understanding of what the nature of that epidemic was, how it was happening, and how to control it. So that's one very good example from the literature. And as with all emerging subjects, there's a lot of issues around quality and quality control. And this is a paper which I would recommend reading, the Croucher paper, which does go into some of the pitfalls and problems of using genome sequencing data in epidemiological studies and how you have to be careful that it's not an exactness, it's not without error, and you need to take that into account when you're doing these analyses. So how are we applying these in New Zealand? Really, we're starting out, but we have got a reasonable large data sets now that we're looking at. We have over 300 genome-sequenced Campylobacter isolates now that we're working on. It's also, when you're doing genomic epidemiology, very important to understand how pathogens evolve in order to interpret the findings. And each pathogen evolves very differently. And Campylobacter is unusual in that it has a very, very high rate of recombination or horizontal gene transfer. So basically, these pathogens evolve via 2 mechanisms. One is single point mutations, picking up mutations through time, and the other is through large-scale movements of large chunks of DNA between different bacterial genomes. And Campylobacter is very, very good at moving large chunks of its genome around between different isolates, called horizontal gene transfer or recombination. And that confuses things, because if you're trying to reconstruct the genealogy of an organism like Campylobacter, each gene, each nucleotide has its own evolutionary history. So there's a lot of work going on at the moment to try and develop techniques which overcome that particular problem. Our first study, which we published a couple of years ago now, was just actually comparing 2 genomes. And these were 2 if you remember, sequence type 474 was the poultry-associated, one particular poultry supplier-associated common human one, and we compared a genome from an isolate from a human case in Manawatu and from a poultry sample taken at exactly the same time, and the same sequence type and the same flagella type as well. And actually, there were striking differences between those two isolates. There were actually 83 genes that differed, and about most of those differences were accounted for by this recombination or horizontal gene transfer. And we could use that to estimate that, probably the common ancestor of these Campylobacter isolates was around about 120 to 130 years ago. And more importantly was an identification of a site called the YKGC gene, which is strongly associated actually with ruminants, as we found out in subsequent infections. So it can be quite useful as a marker for identifying ruminant-associated infection. And others are also doing this as well. Using genome sequencing, we can actually identify many, many more markers associated with particular hosts. So from a source attribution or reservoir attribution point of view, this is very, very useful to get a much better handle, a higher resolution handle on where humans are getting infected from. So we have our YKGC region. There are also other regions identified by Sam Shepard, this vitamin B1 synthesis gene, which is also associated with ruminant infection. It's estimated that Campylobacter jejuni really is only about 4,000 years old. It's been traced back its evolutionary history. It's actually converging with Campylobacter coli, one particular clade of Campylobacter coli, and it's believed that that happened around about the time of onset of agriculture. So that's from a global point of view. But what we can do with our isolates is look what's happened in New Zealand. And we actually have reasonably good estimates of when most cows and sheep and poultry were imported into New Zealand. We know around about 1860, 1870, big numbers of cows and sheep. That's when most of the sheep were imported actually from Australia, and cattle were imported from Australia around about that time, whereas poultry were imported around about the late the 1970s. And the work that we can do by tracing the genealogical history of these isolates that we have from Campylobacter, the common human strain, suggests the common ancestor was probably introduced in ruminants around about the early 19th century. I'd say there's a fair bit of hand-waving that goes on around some of these studies, but if you're really interested in this, it's published in a book chapter which came out in a book recently this year. So that's the kind of work that we can do on Campylobacter. One other piece of work which is very important is if you sample river water across the whole of New Zealand, North and South Island, what you find are Campylobacter jejuni isolates, which appear to come from a cluster or a clade for which, when this paper was published, we had no idea what was the primary reservoir for this particular group. Now, this was a strain of Campylobacter jejuni which actually didn't appear in any of our human infections, so it seemed to be one that was not associated with human infection. We sampled ducks, geese, we sampled swans, we sampled just about every bird, and it wasn't until a summer student, actually a veterinary student, came along and started sampling these birds, the pukeko, that we actually found that this strain is almost 100% of pukekos that we've sampled. And also, in the critically endangered relative of this bird, the takahe, you find this particular strain of Campylobacter is in almost 100% of animals. Now, why is that important? It means that if you take a sample and you're using Campylobacter jejuni as a marker of potential human risk in surface water, then you're going to get a skewed view, because most of the Campylobacters that you isolate from surface water actually come from pukekos or water rails. And when you look at genome sequencing, what you find is that although the water rail-associated strains are potential human pathogens, they have very, very few pathogenic families of genes compared to the other ones which are much more prevalent in human cases. So this is giving us an idea on the reasons why certain strains, although not quite prevalent, don't actually appear in human infections. So lastly, I just want to talk about the salmonellosis epidemic. And this is probably one of the most fascinating ones, and it's really this is work that's ongoing in my laboratory at the moment. In New Zealand, Salmonella kind of oscillates between the second and third most notifiable enteric disease after Campylobacter. It's a complex array of diseases because Salmonellosis is many, many different strains. There's Typhimurium and there's different phage types of Typhimurium. And the one that was very, very prevalent for a long time, caused over 2,500 cases, is this particular strain, Salmonella typhimurium type DT160. And that's the one that I'm going to focus on in the rest of the talk. Now, this one, the first human cases were actually identified in 1998. And there were a very, very small number of cases in and around the Christchurch area. And then there seemed to be a huge die-off of sparrows and silver eyes, so big birds basically dropping out of the sky and around Christchurch. And then that spread very rapidly across the whole of New Zealand and resulted in a 10-year epidemic of this particular strain, resulting in over 2,500 cases. It was the dominant strain for that 10-year period. It's now the second behind a new strain, so it seems to have been replaced by another one. And if you're wondering about sparrows, this is the kind of routes of infection. You can see sparrows and other small birds often feed in cafes. They're also found in supermarkets. You see them quite commonly flying around, and there's even been reports of them using the green-lit mussel beds as water baths overnight. It's quite a common complaint. So you can see plenty of opportunity for transmission. And actually, and the next photograph, I took this at breakfast this morning in the Rendezvous Hotel, and this little sparrow actually came up and started hopping around my table, as if to remind me that I was giving a talk this morning. So as I mentioned, this was the epidemic of DT160, this particular graph here. Peaked around about 2000, 2001, replaced the previous strain, and it's believed that the previous strain actually was susceptible to the phage carried by this strain. So one of the hypotheses is that DT160 carried a phage, phage warfare occurred, it killed off DT130 and replaced it as the dominant strain. And I'm just going to show you how quickly it spread, because this is quite striking. The next slide is just showing how, in green, the DT160 spread from its initial focus around Christchurch area. You can see there's one case around Invercargill here, but around most of the cases at this stage in 2000 were around the Christchurch area. The contrast between a strain which, the 156, which is cattle-associated, stayed mainly in North Island, and another strain, Brandenburg, which stayed mainly in South Island. But you have to look, otherwise you'll miss it, how quickly over the period of just a few weeks, because I'm going to show a 10-year time series, it spread from that initial focus, an explosive spread. So already you can see it spread right the way up to the top of North Island and all the way down to the bottom of South Island. So this was an explosive epidemic that happened over that time period, in contrast to the other two, which tended to maintain their local, stable spatial distribution. Now, we did some typing on these using conventional methods, a whole range of different methods, but unfortunately, most of them had exactly the same pulse field type, the same virulence profile, and so the conventional typing really didn't give us much of a clue about the epidemiology of this infection. So we're now doing genome sequencing. We're doing 120 genomes. We've just done the first 50, and I'll just summarize the results from the first 50 because they're revealing a fascinating insight. One of the insights is that you see the strains from humans, wild birds, and poultry are dotted around there, so it's consistent with zoonotic spread. The second feature is that if you look at the early isolates in the epidemic, they're mainly around the central part of the phylogenetic tree, whereas the more recent ones, around 2009, 2008, 2009, tend to be in the periphery of the tree. And if you use a technique called multidimensional scaling, which just positions those isolates relative to each other on a two-dimensional plane according to their genetic distance. What you see is this picture, which doesn't look very much, but if you then break it down into the years, so this is the early years, 2000 to 2002, what you can see is they're gradually radiating out as we move through time from those central genotypes into the recent years, so those are the most recent years. So basically, as it evolves, it's radiating out. This is consistent with a pathogen, a single source pathogen that probably came into New Zealand, expanded and grew over time. And using some of the coalescent-based modeling techniques, actually, so just showing this in a bit more detail, you can see as the epidemic continued, the genetic dissimilarity between pairs of isolates isolated from each year increases over time. And you can use this in models. This is something called a Bayesian skyline plot, which enables you to model the size of the population of the pathogen over time. And using that information, you can see that there's evidence, strong evidence of a huge, beyond exponential increase in the population of Salmonella DT160 just prior to the big peak in human cases, so suggesting there was a massive amplification occurring, probably in the wildlife population at the time. So that's how we're applying these techniques for Salmonella. One of the main questions we want to ask with this study is how human-human spread versus animals-to-human spread versus human-to-animal spread, the relative proportion of those occurred throughout that epidemic as well, will also inform control strategies. And this is a paper by Alison Mather in Science that appeared last year, which uses exactly the same techniques on a different strain of Salmonella. So just to conclude, how will molecular and genomic epidemiology, or how does it help to inform policy for control? So it helps to inform transmission. I haven't talked about its application to estimating the basic reproduction number, but that was certainly used in flu epidemics. How transmission in social networks, who's infecting whom, zoonotic spread, where transmission routes are coming from, who's infecting whom, and which animals are infecting people, major host switching events that occur over time, useful for source attribution, reservoir attribution, for biosecurity regions, identifying when and where incursions of new pathogens are coming from, and also understanding evolution and how the impact of interventions such as vaccination and antimicrobial use are informing that. And we've got a number of studies on the go at the moment using these genome sequencing tools and techniques to look at a range of different pathogens. So my last slide is just a huge number of people to acknowledge, and just to emphasize again, this is very much a One Health program of research that we're looking at here. It's a strong focus on zoonoses and using a different range of different tools and techniques, and a big team of people. I can't mention everyone in there, but funding obviously from a whole range of different sources as well. So thank you.
John Matthews, that was great. I guess the questions in my mind about Campylobacter were, do you have any information about the host responses? You know, obviously immune responses in terms of some of the data you suggested were age dependent. So I'm just wondering whether that dimension has been looked at all.
Yeah. So in terms of host responses, you mean human host responses rather than animal hosts? Yeah. There's quite a lot of work being done on that in that area around looking at human responses to infection, and certainly it's estimated that in areas, particularly in rural areas, that a kind of constant low dose exposure explains why you see a peak, particularly in young children, and it flattens out in adults, in contrast to urban areas where you tend to see a peak, a smaller peak in children, and then a bigger peak in later in life as well. And it seems to be consistent with this constant low-dose exposure in rural areas and an intermittent high-dose exposure that you get in urban areas. And certainly in other parts of the world where people have done serological surveys, it's consistent with an exposure enough to result in an immune response and a serological conversion around about once every 6 months to Campylobacter in urban areas. I think it's probably more frequent than that in rural areas. We haven't done any serological surveys in New Zealand. I'd love to be able to do that, and I know there are serum banks that could be done to inform how frequently we are actually getting exposed to these pathogens.
Nigel, do you know what was special or is special about the New Zealand poultry industry that resulted in such contamination?
Well, I think it was a whole range of things. I mean, looking at the virulence of that particular strain of Campylobacter, it seems to be one of the strains that comes to the top of the list in terms of virulence markers. So I think it was one thing that was strongly infected. I think a lot of the practices that were being used were suboptimal. They've certainly been improved now, particularly around that spin chiller. Many other countries use air chilling rather than immersion chilling, water chilling, and so there's differences around that. I think there were a whole range of different reasons why our poultry industry were so responsible for such a big epidemic over time. Things have improved, but what I don't think I did mention, I'm not sure I've got it on the next slide either, but we've gone from being the worst in the world to the second worst in the world, now behind the Czech Republic. So I didn't want to give any impression that we should be complacent. It's not just because I want to keep the research funding coming in, but it's extremely important that we don't lose focus on poultry as a source of infection in New Zealand.
Well, Thanks, Nigel. I think it was an absolutely outstanding presentation. We're very pleased to continue the One Health theme, and Dr. Joanna McKenzie will now be speaking about strengthening epidemiological capacity with the One Health framework. She's a senior lecturer in the One Health Epidemiology and International Development at Massey University, and she's leading the academic program for One Health Epidemiology across Southeast Asia. She has a very broad range of experiences from clinical veterinary practice through managing surveillance and epidemiological studies for the Ministry of Agriculture and Fisheries, and really over the last 12 years has had an international focus, particularly in Asia and the Pacific region, on strengthening biosecurity and surveillance capacity. So over to Joanna.
Thank you very much, Michael, and good morning, everybody. It's a real pleasure to be here and to have this opportunity to present this morning. I think in the spirit of One Health, it's a fantastic opportunity to build relationships between the medical epidemiology and veterinary epidemiology sectors in New Zealand. So thanks for the organisers for setting this up. So Nigel gave a nice introduction at the beginning of his presentation on the importance of zoonotic diseases as sources of pandemics and also of existing neglected zoonotic diseases such as rabies and brucellosis. And so I'm going to talk today about some work that we've been doing at Massey University in building epidemiology or strengthening capacity in epidemiology and biosecurity in the framework of a One Health context. So doctors and vets, wildlife health specialists working together in South Asia to improve skills, understanding of methodologies and relationships to detect potential pandemics and to manage zoonotic diseases. So my talk this morning has been prepared by myself and Peter Jolly, but it actually, the work that I present involves the input of a huge number of people. In particular, the IVABs at Massey University and the Centre for Public Health Research at Massey have been the sort of key partners in the academic parts of the program, and then wider collaboration with National Otago University, international organisations, London School of Hygiene, Tropical Medicine, Tufts and Prince Edward Island, a whole range around the world of input. So this represents the inputs of a lot of people. So today I'm going to present on the approach, our education into action approach. I'll go through the components of our One Health programs, and in the context of the theme of the conference, Epidemiology in the Age of the Internet, I'll outline the internet-based technologies that have really helped, or that have been essential to delivering these programs, and the key outcomes that have arisen, and at the end I've just added a few other examples of technologies that are using the internet. So the fundamental aspect of our approach is working with human health, domestic animal health and wildlife and environmental health professionals, integrating education and with applied training and supporting this with organisational development to support collaboration. So the fundamental approach is an education into action approach. So beginning with education, we have set up at Massey an integrated One Health Master's degree program, which involves a combination of 2 course-based master's degrees. And the first 4 papers of each degree, all people from all professions study together in the 4 sort of core building epidemiology, sort of fundamental epidemiology papers, and after that specialise in a range of papers that provide the opportunity to specialise in particular aspects relevant to their own discipline or applications of epidemiology methods to their own disciplines. And the vets and the doctors graduate with a Master of Public Health and Biosecurity, and not the vets and doctors, the doctors and the public health professionals, and the vets graduate with a Master of Veterinary Medicine. So this is the education aspect. It really strengthens the individuals through their motivation to study and learn, and builds strong relationships between individuals as they study together. Following on from this, or integrating with the education side, is the applied training side, which is really built around what we've called collaborative investigation projects, which involve people from different sectors working together to implement epidemiology projects on zoonotic diseases of importance in their countries. So working together to design and budget their projects, then in the field collecting samples and questionnaire data together, and there's some photos of team working in Afghanistan, and then organising and supporting these projects with mentors from around the world to help with designing, and running workshops where the participants can work with their own data and working on data management and data analysis and interpreting the results. So together, this is the applied or the action side, which moves more into strengthening at the institutional level, in addition to strengthening individuals, but more to the institution, where it incorporates a wider group of people than those that were studying in the degrees. It involves the different field, national, laboratory, and it involves different institutions working together. It also involves government institutional permissions and engaging at that level. So this applied work has been particularly valuable in building relationships between the institutions. And then wrapping around the education and action is developing an organisational framework that supports collaboration, which we call One Health Hubs, and I'll go into those in a little bit more detail a bit further on, but I just mention them at the moment. So those are the key elements of the approach, and using different combinations of those, we've been involved in 3 programs now delivering 3 One Health epidemiology programs in Asia. The initial one was a regional program for building epidemiology and human health, animal health capacity in South Asia, in 7 countries of South Asia, funded through by the European Union through the Avian and Human Influenza Trust Fund, which was administered by the World Bank. This was a 4-year program in the 7 countries shown there on the left. And the master's degree involved 68 students with a good range between public health and animal health. And then as phase 2 was the applied stage, and it expanded to include about 150 people involved in the projects. Then following on from this, the master's degree was then also run with a cohort from China and Mongolia, a cohort of 19 with 9 public health and 10 animal health students studying, and that project was just the master's degree and didn't include the applied stage. And now next week actually, we're just about to begin a 3rd program funded through the EU, which is a One Health in Asia program, which will be, it's a 3-year program in 4 countries, in Afghanistan, Bangladesh, Bhutan and Nepal. And the core of that is a fellowship program where we have 24 fellows. So we've got 10 from the public health, 8 from the domestic animal health, and 6 wildlife health veterinarians from the 4 countries. And they'll study the master's degree, applied projects, and will also develop training materials in epidemiology in their own national languages to carry on sort of training beyond the end of the program that we're involved with. So I'll give you some more detail on the regional training in animal and human health epidemiology program in South Asia, which was a real journey over 4 years. It began with the master's degree, which is the formal training, which ran for about 17 months in the 2010 through to 2011. Following on from that, we then worked in the countries, had a series of workshops to identify priority diseases and needs assessment, so needs for facilitating One Health, and also what were the priority zoonoses, what were the gaps in information that could be filled through project work. And then had a number of mentors around the world who worked with different groups in each country to establish their collaborative investigation projects, and these projects involved or created an opportunity for applied epidemiology training to get experience working together in collaborative projects. Also very valuable experience in project management and producing some useful zoonotic disease information. So once the the data had been collected, we ran a series of workshops with in-country and regional workshops to support in managing their data and using sort of as much as possible open source software, EpiInfo, and basic descriptive analyses, and then some more advanced analytical workshops. Towards or around the same time, worked with the countries to develop One Health hubs. And then towards the end of the program, facilitated a national symposium in each country where the results from the projects could be presented. Each country produced a set of resolutions for how they could grow the One Health approach in their country and strengthen their One Health hub. And the grand finale was a regional symposium where participants from all the countries came together. They could present their results. There were regional discussions on zoonotic diseases based around the results, a simulation exercise, and produced a set of regional One Health resolutions. And this really worked to strengthen the One Health network. So a long journey with many aspects to it. So I'll just go into a little more detail about the components and how these were supported, sort of the technologies that supported these. So as I said, the master's degree involved students from public health backgrounds, domestic animal health backgrounds, and in this latest program, wildlife health backgrounds, studying together and working together in the first 4 papers and specialty papers. This program has been delivered, it's through a blended learning approach, so predominantly online and involving delivery of the course material through the learning stream, which is Massey University's learning management system, and interspersing this with 2 or 3 workshops where the participants from the different countries all came together. So Stream is a wonderful system for teaching online and creating a sort of a classroom environment online, and you know, courses can be delivered and involve a range of a range of different softwares. Material is presented and then interspersed with assessments, and some of these involve group work, so that we've set up groups with different sectors, different professions, working together from different parts of the country, all working together online to design a disease surveillance program or to evaluate disease control options and these sorts of things. So particularly the group work, which was challenging to set up, challenging to assess, but strongly facilitated the relationship side. And then also learning through online scenario and unfolding disease outbreak, where the students were the investigator and had to do a outbreak investigation, gather data, summarise and analyse that data, work in groups to discuss their hypotheses, and to, at the end, to actually write a report. So it was a very realistic environment where they stepped through stages and had a number of different learning aspects to it in terms of outbreak investigation, interpreting data, testing hypotheses, and writing a report. So the advantages of the internet in the master's program is that it's really enabled delivery of these courses to large numbers of students globally. So in this case, we had 68 students in South Asia, And the sort of critical mass really adds to the value of the program, in that it's I guess, builds a big alumni, and who are very proud to be alumni of Massey. And so that when they get together at meetings in the region and so on, they meet each other, and there's already a bond together, and a common goal of strengthening One Health. It's meant that people or the students can remain in their country while they're studying, which is a lot cheaper. It also means that there's less, they can stay in their work, and this has been particularly important for the senior managers. So in the first program, a number of senior managers actually took the opportunity to do a degree because they didn't have time to leave their job. It means you can have input from teaching people anywhere around the world, and it also means it can facilitate group work with different people from different sectors around different countries. There's obviously a number of challenges, and particularly through internet access in Nepal, they have up to 18 hours of load shedding or power cuts a day, and often 3 o'clock in the morning is the time when the power's on, so they're getting up then, doing their washing and doing their studying at 3 a.m. in the morning. And it's truly humbling that how committed and how a number of these students actually got through their degrees. In Afghanistan, there was very low bandwidth, and loading stream would take 10 minutes, so they'd turn on their computers, load stream, and go and have a shower and come back and start their studies. But they still succeeded despite all these challenges. It's just a shot here, which I love, in Dhaka in Bangladesh, of just this wires of that run up into the offices, and then they're all sort of coiled around, and then you see them disappearing up into another office, and that's the internet connection with the rest of the world. Communication was challenging through being remote. Like when in teaching, if somebody wasn't responding and not submitting their assignments, you didn't know if it was because they weren't understanding, or if they were away, or if they... and quite often it turned out that they were busy and they were away on workshops or work trips, but they didn't think to let us know where they were going, and so the gap in communication can be quite strong. Obviously, challenges in combining study and work commitments, and as in any study, quite a range of backgrounds and abilities to work with. And the periodic face-to-face was a really important part of building up the relationships and the group spirit as well. So moving on to the collaborative investigation projects, in this South Asia project, there were 13 projects that were designed across the 7 countries that provided the opportunity for epidemiological skills to be applied to study design, to implementation aspects, going out sampling, how to manage non-response, analysing data, reporting, and some actually had some contribution to produce some data that could be of value to policy evaluation. And they also provided a focus for international expert assistance input and providing further operational training. So the studies, there were zoonotic diseases in the different countries. Some countries, some diseases were studied in more than one country. An interesting study in Bhutan where they actually did an evaluation of how effectively the animal health and the human health sectors communicated when there was a suspect zoonotic disease outbreak, who communicated with whom. Interestingly, they found that the public health sector was more proactive in communicating zoonotic disease outbreaks than the animal health sector, which surprised everybody because the vets were thinking that they were the ones that were more proactive in One Health. But it produced some very nice information to help to develop further One Health in Bhutan. Just a quick show. So the people that were most closely involved in the projects, we had about 150 people. Then we're expanding out to those who were trained and were collecting data, expanded out to about 1,700 people in Bangladesh. In one project here, they trained over 1,000 people to collect data on cases and controls in villages in Bangladesh. And then if looking at people who were involved in subjects in the studies, over 7,000. So big scale. The One Health hubs, it was an important component of the collaborative investigation projects, provided an important focus or a useful focus to establish One Health hubs. And a lot of this came by engaging the ministries, the Departments of Animal Health and the Departments of Public Health to get permissions for the projects, to get the funding in place, to go out and collect the data. So they had to agree to work together, and that took, in many cases, a lot of groundwork. And being good Kiwis, we just sort of rock up and knock on the door and just sit down and have a chat and sort of get people together. And as over time we built up relationships and built up support for One Health in the country, sort of built around the collaborative investigation projects. So this was the organisational framework. It was involved the two key government departments as the foundation organisations, and who are leading it and who are managing the members of the hub and the direction that it's taking. So it's very much embedded in the government sector, but with engagement of NGOs and universities and some international agencies. Creating a functional network to facilitate communication on One Health and to share information and resources, and also to facilitate collaboration in new projects. And then the One Health hub in each of the 7 countries was linked to form a regional One Health network. And so underpinning the collaborative investigation projects, the One Health hubs and the regional network is a web-based system called HubNet. So for the master's program, Stream was the key sort of technology. For the applied part of the program, HubNet was the key supporting technology, together with the usual email and Skype and cell phones. HubNet is a system that a web-based system that we've developed through this project to support the collaborative projects, One Health hubs and the regional network. It includes powerful web-based communication and collaboration tools, and it's been built in open source software. The idea is ultimately that it would be handed over and used in the region, and so we've built it around open source software as much as possible, with a few sort of modifications. And it enables people who are in just different countries and different buildings and different cities and different provinces to work together, and to also get input from people around the world, and as a repository for information of use across the different sectors. So HubNet it's web-based. It has a public, so there's 3 areas. On the website, there's the public, which anybody can see. Then when logging in, it's another Drupal page, which the members of the One Health network can see news and can see blogs that have been put into the network site. And each of the One Health hubs has its own page and is represented here, and can put up articles to show off what they're doing in terms of One Health work and project work. And we're just in the process of setting up now that they'll be able to write articles and publish articles from their site out to the front page, with going through a sort of permission process, so that there's some control over what information the hub publishes. And then what we call on the inside, which is like the office space, I guess, is groups. It's set up with a number of, as many as you like, different secure sites or groups of people with restricted membership or membership with permission. And then those sites can be used for sharing, for a repository of documents, for developing things like a project proposal together, version control, for updating on news through the blog sites, and sharing on information that's been useful. So this is just a little example in the Bangladesh One Health Hub. There was a blog about anthrax cases put up by the public health sector, and then they also added a graph showing the timeline and temporal distribution of cases in different regions. So it's a slow process of getting these going, but as we can encourage that sort of information, it creates the opportunity for sharing for awareness between the different sectors. In each country, we organised a national, or supported the organisation of a national symposium, and so altogether had about 700 delegates across the 7 countries. And these symposia were organised by the One Health Hub in each country. They were the opportunity for the participants in our project to present the results of their projects, but also bringing in lots of other projects that were involved in One Health in the country. So it was a real integration of this activities in this project with the wider One Health activities in each country. And it also provided the opportunity to engage with international agencies and organisations like Wildlife Conservation Society and the International Centre for Diarrheal Disease Research in Bangladesh, and strengthen the relationships in each country. And the grand finale was a symposium in Bhutan in the end of 2013, where we had 121 delegates from the SAARC countries, and we had a mix of the technical specialists who'd been involved in this program and more senior people from the Ministry of Health and the Ministry of Agriculture, together with international speakers and facilitators and representatives from the World Bank and the International Agency. So it was created a real dynamic environment, and brought together the workers, the epidemiologists, and the decision makers, and that was particularly valuable in a disease simulation exercise, which was called Mysteria, which was a disease that was a fake disease that broke out in an area in Bhutan and involved cattle and horses and people and spread to different countries. And so it was very interesting to see the dynamics between the epidemiologists and their decision makers, and in which directions, who was heard and which directions they were taking. A key outcome of this was a commitment to collaboration of a One Health agenda in the region. And one of some symbolic events, symbolic acts, I guess, can be quite powerful. So we facilitated the development of a regional set of One Health resolutions that all countries agreed to. That was to progress One Health in the region. And the Vice-Chancellor Massey was there, and we had it in a scroll, and he handed it to the senior most person in health and livestock in each country, and sort of handed over the baton of their responsibility for now progressing One Health in their region. And we visited Bhutan earlier this year, and the Director of Livestock was very proudly showing us that he had his PARO resolutions up on his wall, and he looked at that every day, and he was very committed to carrying on. So key outcomes from the project: there was a very high level of engagement starting with the master's degree and then through the collaborative investigation projects and the One Health hubs. And the project and program provided the opportunity for hands-on experience with the One Health approach, which was very valuable, and provided, I'm going to give a presentation a little bit later, more on the relationship side there, but provided a real opportunity to build relationships and get confidence of working with the different sectors. The projects were able to demonstrate the utility of the One Health approach, and particularly in collecting samples and data from people and animals in the same household, and being able to link the two more strongly. They also provided a focus for establishing the national and regional One Health networks. The activities certainly strengthen relationships and understanding, confidence and trust between the sectors and the countries in the region through all their various interactions. And they started out, quite, especially a lot of the government departments and senior people were quite hesitant, and then as they got engaged and saw the outcomes, we're very proud to then be involved with the outcomes of the project. And that was a value of the regional symposium, is that they could all come together and show off what they'd been involved with. So the government decision makers did become a lot more engaged in One Health, and a lot of that was the way we worked with them to integrate it with what was already happening in their country and happening. And following the program, Bhutan and Nepal are both developing One Health national One Health strategies. The internet was central to being able to deliver a multi-sectoral and a multinational education interaction program across the 7 countries, which was managed from New Zealand and with input from people all around the world, and to be able to create and to maintain the One Health networks. Just quickly, I'm not sure how I'm going for time, but okay. So just in addition to the One Health programs in Asia, I just wanted to give another couple of examples of some software, the software that's been developed at Massey, which is really linked into the internet theme of the conference. So there's Incident Iris, or the Incident Response Information System, is a web-based, or is a very flexible database structure that data can be entered through the web anywhere where you have access to the web from multiple sources, and it can manage disease outbreak data from different species, different areas, and also in different languages. So it's a powerful database, and it's underpinning a suite of tools called HandyPlan, which are a range of tools for integrating data from IRIS, for integrating different geographical layers, environmental layers, population distribution layers, climate layers, and using that data to develop risk surfaces for targeting surveillance, and also risk surfaces for distribution of endemic disease, and integrating it with modelling and evaluating disease control strategies. So it's spatially got spatial capability and can map outbreaks and conduct some spatial analysis like kernel smoothing to look at highest density areas of, this is avian influenza in the Mekong area in Vietnam, to combine different layers of data to create risk services. I think this is actually a layer of cattle distribution, cattle population density in Sri Lanka, which could be combined with environmental data to look at, say, risks of vector-based diseases. A very cool product that's been developed through the Public Health Foundation for India is the Swasthya Slate. This is just a little box that is carried around by health workers as they visit village people. And it's got capability for about over 33 tests, and is connected to a database through the cell phone network. So health workers can be visiting villages, they can be collecting data and parameters from the patients, they can do photographs, and they can, it's got a GPS for location, they can get some questionnaire data. So it creates an opportunity to a powerful opportunity to collect data from a wide range of sources that could be used more in epidemiological studies. So that was there's a lot of potential with tools like that, and I think there's a bit of a similar tool being used in Northland in New Zealand in the Maori communities. So to end, I'd just like to acknowledge the European Union and the Avian and Human Influenza Trust Fund for their support for the funds in the World Bank, the governments of all of the countries, Massey, and then the multiple collaborating organisations, which I won't list individually, but thank you very much. It's covered by the funding, because there's not too many students that could afford to pay the international fees, so it's very much dependent on having funding from the international agencies. And one of the, I guess, an advantage of doing it that way is creating cohorts of students that study together, which has been a real strength.
John Matthews. Could you comment on the interface with other organisations such as WHO in this international health space?
Yeah, so the collaborative investigation projects provided an opportunity to interface with the international agencies as much as possible. So we met with them, engaged them, discussed what we were doing, looked at opportunities for how they could be involved. They were involved to varying degrees in different countries depending on the diseases being studied and the interests of the people involved. In Sri Lanka in the rabies project, WHO was involved in the design of that project. Then probably not so much involved in the projects themselves, but came together in the National One Health Symposia. They were contributing in there and also at the regional symposium. So they sort of, I guess by bringing them in, FAO and WHO in particular, and their being aware of what was happening and connected with what was happening enabled them to, I guess, provide some additional sort of support and encouragement. So probably not so much as a integral hands-on, but just by general support and engagement.
Annette Dobson from University of Queensland. This reminds me very much of the International Clinical Epidemiology Network that was funded by Rockefeller in the late 80s and early 90s. It has so many parallels. And I wonder what you have planned for the long-term sustainability, because the awful thing with INCLEN is that there's very little institutional development that's been sustained. Obviously, we train some superb people who are in very important positions, but the sustainability, I think, is incredibly important, and if it could be built at the early stages, that would be fabulous. Yes, you're absolutely right, and we've been very aware of trying to create a sustainable, or the environment to sustain the initiatives as much as possible. I mean, one of the challenges is the high turnover of people in key positions in the government. So you get somebody who's a supporter and really involved in One Health, and then they're moved on, and the next person, they've got a completely different focus, a clinical focus or a surgical focus or something. So one of our rationales of engaging the government and working through the government, which has a lot of challenges, but was to actually strengthen them and bring them into a framework, and as much as possible to strengthen the initiatives that they were taking themselves. I mean, Bangladesh is a leader in developing a One Health strategic framework, and has, with support of FAO and WHO, and has got their ministers to sign it and support it. It's still a framework. They're still just, after 5 years or so, just sort of looking at setting up a secretariat. So there's a very long time frame for these things to evolve over time, and I guess in that situation, luckily the two leaders are still in key positions and can keep things going. I guess as much as possible, we've sort of been trying to support the in-country leaders who are active. To be realistic, it's some initiatives will keep going in the countries, but it's still largely catalysed, I guess, by projects, and where there is funding to do projects and to inject some enthusiasm and activity. And with the One Health HubNet and the One Health network, there's about 200 people from South Asia that are signed up to the One Health network, but actually getting them to engage and contribute to it is a slow process, and luckily we have another program that'll carry on now for another 3 years. So through that we can continue to build the
link in what happens and the people involved in this current project with what's gone before and try and build on that. And also then in this particular project coming up now has a training the trainers component to it, where they'll be trained using the epidemiology materials and experience so that they can develop training programs for in-service training or undergraduate training. So trying to find opportunities, but yeah.
Kia ora, thank you for your presentation. I'm quite interested in the software package that you used for the online learning and ongoing communication. So my questions are, what did you use? How did you decide that it was the most appropriate? And what were some of the pros and cons with the particular package that you used?
Okay, I could probably answer some of that, but not all of that. So the online learning system is Massey University's learning management system, which is used throughout the university. So we basically were users of that, not developers of it. But it was it is a very effective tool for delivering training, and it's got good communication and good ways of linking into other resources. The weakness of the online learning system, which meant that it sort of didn't carry over into the project side, was that you can't it's more limited. Well, you have to be registered at Massey for one thing to be a part of it, and it's more difficult to form groups and have groups operating. So a major deciding factor influencing HubNet was software that could enable the establishment of groups of people. So HubNet has a very flat surface in that you can't have a hierarchical grouping. You can't have sort of South Asia region and then Nepal, Bangladesh, One Health hubs, and then projects within it. You just have, if you think of lots of different circles, and each of those is a group, and they the members of that group have to have permission to become in that group, so it's secure. And so that you can have a smaller group, you and I could set up a group to if we're working on a project, or we could have a AEA group that could share information on the across the sectors on epidemiology. So HubNet, I guess the particular strength of HubNet was this ability to set up groups, and through the Alfresco software, and then because that was very much a sort of a workroom type environment, and it wasn't very appealing in a lot of ways, didn't have a lot of eye appeal to people. They had to log in and go in and find their site and so on. We set up a Drupal-based front end, which was where when they came into the website, they could see news, they could see a range of activities, they can put out news there, and without even having to log into their various sites. So that's, I think, been a really useful tool for sharing information and news and attracting people into the software.
We'll finish there, and just a big thank you to Joanna for that remarkable presentation.